Okay, so today we're going to be talking about base pairing / protein synthesis. So first off, we'll start off with what is protein synthesis. So protein synthesis is the process in which proteins are created. So the mRNA must be transcribed from the DNA, and that is transcription, and then translation is where the mRNA is converted into the actual protein. That's the central dogma of biology, DNA to RNA to protein. And then the base pairing rule is used to create a complementary RNA strand, which is the transcription part of it, and then it goes to translation, which is the protein. So we have 4 nucleic bases in DNA, and these are adenine, guanine, cytosine, and thymine. And in RNA, the thymine is replaced with uracil. So in DNA, however, we're going to see the adenine pair with thymine and the guanine pair with cytosine. So again, like I was talking about, these are the base pairing rules. Adenine pairs with thymine, but in RNA, the thymine is replaced with uracil. So just in RNA, adenine is going to pair with uracil, and guanine pairs with cytosine. Then to find the amino acid sequence, we look at the DNA strand, we find the complementary mRNA strand, and then use a codon chart to find the corresponding amino acid. So for example, this is the DNA strand we have here, and then the mRNA strand, we're just going to find the bases that are going to match with them. So we look at the beginning, we see guanine, and we know that cytosine pairs with guanine, so that's going to be the first one. And then since this is RNA, we are not going to have thymine, and we are going to have uracil instead. So for the pair of the adenine, we're going to have uracil, and then the thymine pairs with adenine, the cytosine pairs with guanine, etc., etc. We keep going until we have the whole mRNA strand. And then we want to take them and split them into threes. So these group of 3 bases is called codons, and these codons are going to be really important when we're trying to find what the amino acids are. So this right here is a codon chart. So you start at the middle, you find the first base in your codon, go to that quadrant, and then you find the second, and that's going to be one of those 4 sections, and then you go to your third base, find that, and then you're going to have your amino acid. So, for example, if we look at the strand that we had earlier, we can do it for the sake of, so we're going to take the first codon, which is CUA. So we go back here, we are going to have this third quadrant, right here, the fourth quadrant right here. So it's going to be C, and then we go here because the next one is going to be U, and then the third base is A. So our amino acid is going to be leucine. We just do that for all of them, and then when we look at our last codon, this is a stop codon. So when we get to one of these, so UGA stop, this is where the polypeptide chain breaks down and the proteins are stopped produced. So here we just have some review questions. Take about 30 seconds to answer these. Okay, so this first question, it's kind of working backwards because the example that we had, we went from DNA to mRNA, but it's pretty much the same. So you just find the corresponding pair, and remember, wherever you see an adenine, it's going to pair with a uracil. You see a uracil pairs with adenine. DNA to mRNA in the second question is very similar to the example that we had. And then the third question is just completing all of the steps. And so um, and then we use the amino acid chart and codon chart, and we find the amino acids.